So the paper we're going to be talking about today is the crystal structure of the S-adenosyl or methionine-dependent mycoic acid synthase UmaA from Mycobacterium tuberculosis. And that paper was we have two authors here today, Sean Tong and Jia Wang. So I'll have Sean first introduce himself.
Hi, my name is Sean Tang. I am the author of the paper. I did this paper in 2022 when I was in WashU, and we were primarily working with elucidating the structure of UmaA and doing a bunch of computational analysis, and we ended up writing a good chunk of the results.
Right. Jia?
Hi, my name is Gia Wang. I worked with Sean on the analysis portion of this paper back in 2022 at WashU.
All right, great. So I'm just going to start off with some sort of simple questions. So tell me a little bit about yourself, Sean. Tell me a little bit about your background and essentially then what led you to choose the bioinformatics course that I teach. So a little bit of background here. I teach a course in structured bioinformatics at Washington University, and Sean and Jaya both took my class, and this paper was a result of that class. And so, Sean, if you can tell me a little bit about yourself and kind of what made you choose this class over the other classes you could have taken at Washington University, or this particular course.
All right. Well, I came to WashU first studying psychology and neuroscience. I wanted to go in that route, but then I eventually took organic chemistry, and I really fell in love with the class. I just looking at those molecular mechanisms just made me realize something like really profound about the world, and so I got really, and instead of studying psychology and neuroscience, I got into biochemistry. And so I decided to switch to major in biochemistry, and I found this class called structural bioinformatics, and it was really fascinating because you're it's all using molecular visualization software, and I just thought, wow, just being able to see uh, the molecules in 3D firsthand with your own eyes is just really, really amazing. And so I decided to take a look into the class, and I really wanted to get into that.
All right, cool. Gia?
Yeah, so I came into WashU. I was just a bio major, and I hadn't had a lot of um, experience where I was able to apply what I was learning in the classroom to anything that was, research or like in real life. And I think so. I heard about this class from one of my friends who had been a TA for the class, and he told me about how he had actually written a paper and used all these really new technologies to study like a specific protein. And I thought that was just so cool. And I honestly had no idea what to expect going into the class because I had obviously never done anything like that before. And it's overwhelming to think about how you go from just learning about just simple like organic chemistry or just the amino acids, and then you go into a class and you're using your knowledge to apply it. So I decided to take it really from his recommendation and how much he really liked the class, and then I got so much more out of it than I would have thought, and definitely just a lot more expanded understanding of, I think, biochemistry and science.
Okay, so would it be fair to say then your experience of protein structures was small before taking this class, Gia?
Oh, yes, definitely. I mean, pretty much just, learned about them in class. That's it.
Right, how about you, Sean?
Oh yeah, much smaller. Yeah, this class really got me interested into drug discovery, and so it's something I want to pursue in the future now.
Okay. And also, have any of you, and I go back to Sean with this question, have you done the research in a lab before this? You had any lab experience before taking this course? Were you working at undergraduate, doing some undergraduate research?
Yes, I worked in a cornfield lab just a summer of, and it was mostly it was pretty much only a wet lab.
Hmm.
So being able to diversify my experience with dry lab doing computational research was very rewarding.
Good. How about you, Gia?
Yeah, similar to Sean, I had only done some wet lab research in the past, and it was nothing like this project where, obviously we didn't do the purification and everything, but we did the analysis portion is what we worked on. I think as an undergrad, um, I learned a lot of lab techniques and I learned, some basics of analysis. But in the lab that I'd done before, we worked with just knockout mice, and really the stuff I was doing was helping with flow cytometry and things, just more technical things. And so I
Hmm.
think doing the analysis was very different for me.
Okay, cool. So once you received your science structure for my class, what were your first steps to begin your analysis? What were first things you did? Actually, first, now I'll go to Sean with this. Were you overwhelmed or were you excited? So what was that first sort of visceral response? And then what were some of the things that you started doing?
I would say I was excited, and a little also a little perplexed with the structure because and the name of the structure is unknown mycolic acid synthase. His keyword is unknown, so there's not much known about it, and so that one was all. So there's a lot of potential with it. Then um, either there's nothing much known about it, so you're gonna have to dig a lot into the research and try to find something, or you're gonna have to try to do something very creative then. And so I just saw a lot of potential with it, and so I guess it's a both excitement and overwhelming.
Okay. Gia?
Yeah, I think me too. Definitely like given a protein called unknown, but I think because of the resources in the class and like, we were kind of just told that, we start with the resources that we had, and luckily it is part of a family of many like of these like methyltransferases, MMA, PCA, all of those. So reading up on the literature kind of gave us a lot to work with, um and kind of like gave us a start, I guess.
Mhm. So you say the start would be then doing a literature search. Once you get your protein, go into literature and saying, okay, where does my protein fit with what's out there in literature, right? Doing some simple literature searches, right? So it probably was a monumentous task, right?
Mhm.
But you sort of take it one bite at a time. And so would you agree the literature search would be the first thing to do, Gia?
Yes, I think, yeah, because we really didn't know much about, I mean, I meant like sorry, UMA, or just, I mean, honestly, like in tuberculosis, in the first place. And starting with literature kind of gave us just more background about, the importance of why it's important to study it, first of all, and then just a general understanding of what it does and
Hmm.
where to start.
Sean?
Yeah, I would say the same thing. Just doing the literature search gave us a good stepping stone into the project. Doing a lot of background research in the metabolic pathway where the protein lies, and also the where it lies in relation to its relative related proteins, the homologs. And so those were very useful, just studying the homologs as well.
Okay. So what were some of the computational tools or databases you used, if you can remember, that you used or you liked, or you can comment on some of the stuff that you used to determine the structure? Since this was a computational course, you weren't doing wet lab stuff. So what are some of the computational things that you did? And we'll start with Sean and then go to Gia.
We can't go without mentioning Primal. Primal is the most important one. Um, but we also used a lot of software online. This included PDB, PDB sum.
Hmm.
Another one was Cluster Omega. that one was important for the bioinformatics. Yes, CBDoc. Yeah, CBDoc was important, and also AlphaFold. AlphaFold just came out that really it just came out that semester, and so it was open to the public, and so we, since it was new, we just took advantage of it and tried to push the boundaries on what this class could do.
Yeah, actually, I think you may have been the first group, your team, you and Jim may have been the first two people in my class to actually use AlphaFold. So you're the beginning of all that. So is there anything else you want to add to what Sean said, Gia?
I think that pretty much covered it. I think we used Dolly to do some
Hmm.
alignments, but I think that covers it. Yeah, I think it's very, there were just so many resources, like, especially now, and I bet, I mean, more are coming out, so.
Hmm. Yeah, and AlphaFold's gotten a lot better since
Mhm.
2022, and it's only going to get better. And a lot of the AI tools out there, are starting to, and now AI is getting combined with all these tools. So we're going to be seeing some really, I think, interesting things come out in the next several years. And this one's going to go to Gia. So what were some of the unexpected features or challenges in the structure that you found? Was there something interesting or challenging you found about the structure?
Yeah, that's a good question. I guess the one that comes to mind right now is like for example, when we first saw the structure, there's stuff bound to it, like a nitrate, for example. And we just kind of didn't talk about it much, or we're not really sure where it came from, especially I think not having ever done anything like with the purification process. We
Mm-hmm.
just, a lot of times we would just be like, oh, like, so it must have been from the purification. And then later on in our research, I think just like after talking to more people or after getting like our feedback, it's like, oh, like, but there wasn't any nitrate. And I think it was just confusing for us, especially um working with these softwares for the first time and then kind of like doing an analysis for the first time, like the assumptions that we think we can make and then not really knowing what actually can be made as an assumption or not was I think that was challenging for us.
Yeah. Yep. Anything you want to add, Sean?
Well, I guess another uh, confusing part of our project was the studying the N-terminus of our protein. It was different from the other homologs. This one was sticking out into the solution, whereas the other ones had an N-terminus that was more closed into the protein. And also, when we put it in alpha fold, it gave us similar structure to the homologs. So in this case, we tried thinking about like a, a solution to why it could have be in that situation. One of it could just be that it has a his tag on, and maybe that's why it's
Hmm.
out in the solution. Another one is maybe proposing like sort of like a mechanism. Maybe it's something to do with how the protein functions. The N-terminus happens to be right over the active site, and so we propose maybe that it's acting as like a gate where it can be an open conformation and a closed conformation, and it can allow the substrates to enter the active site and a product to leave the active site.
Yeah, no, that was a very interesting part of the paper. Very interesting observation. Actually, I think you guys made two really interesting observations. The one about the nitrate, and we still haven't figured out where that nitrate came from. To be, it just appeared, but it's somewhere through the purification process. We didn't actually add any nitrate anywhere in the purification process, but it ended up there, and that nitrate. It's isosteric to carbonate, right? And so that certainly, when you look in literature, there are several papers that have, the carbonate as part of the mechanism, right? And what's also interesting is that if you have a glycine at that position, you need the carbonate as part of the mechanism. And if that residue then is a glutamic acid, which kind of looks like carbonate, you don't need it. So you become bicarbonate dependent by that sort of glycine to glutamate's mutation. And so that was a little cute little observation you guys figured out there, which is very nice. And so, so when you're writing your paper, um, what aspects of the paper that you found most challenging? I'll ask Gia that question and then ask Sean the question too, but go Gia first.
Um, that's a good question. I feel like there were many challenging aspects for me. Um, I guess like one really challenging aspect for me is just like kind of translating that classroom knowledge into this paper. Because obviously I had taken like biochemistry and organic chemistry in the past, but like I I just think the level of it, like when you apply it to this paper, was just at a much higher level than I had ever been used to. Like whenever we were thinking about different mechanisms or how possibly like for example, just with the bicarbonate and the glycine versus like the glutamate, it's just like thinking about just like how the structures would fit or how just the charges would work in comparison, let's say, with like MMA A4 or PCA. Like I think putting all of that together was very hard for me.
Hmm. Okay. About you, how about you, Sean?
Yeah, so I'd say the probably the most challenging part of writing the paper was uh trying to not make it sound dry, especially when it goes to structural biology and you're talking about interactions. Uh, you could start getting to the point where you're listing amino acid residues and atoms of the amino acid residues, and it could just
Hmm.
sound like a bunch of letters and uh, and words, amino acid residues. So it gets really dry. So um, I it was challenging to make it sound like a story, and so but I thought trying to make it sound like a story would be the way to go, and so
Hmm.
I tried relating it to other parts of the paper. Like for example, when I mentioned some amino acid residue like glycine interacting with S-adenosylhomocysteine, I'd go back and relate it to a part I mentioned in a previous section about this GXGXG motif, and
Hmm.
bring that back and say, well, this glycine happens to be in the GXGXG motif, which suggests how important this motif is for binding to
Hmm.
S-adenosylhomocysteine.
Okay, cool. No, that's yeah, very interesting. One thing I think which interesting part about this particular course is that it sort of it kind of you have to bring all your information to bear that you've learned before. It's not just biology. It's not just biology and chemistry, organic chemistry. It's all these things, right? And so you have to. There's a little, in particular with your structure, you had to know a little bit about kinetics. You had to know a little bit about biology. You had to know a little bit about biochemistry. You know, had to be able to a little bit about the mechanism, certainly proteins, all these things come to bear. All that stuff you've been learning the first few years, your sort of the first couple of years of your undergraduate career. So you need all those things, right? When you agree to do this, it's not, and that's what this integration of information is certainly where, and this is where you start becoming a mature scientist, right? Is learning is taking all this information that we've given you in these sort of introductory courses, and then now using all that to look at a particular research problem, right?
Right. Yeah, I
Yeah.
would agree with that. I'd say that's probably one of the most rewarding aspects of the class. Um, and just like applying all the things that we've learned and putting it into this exploratory discovery process makes this class very, very amazing.
And so what another part of the course is the peer review process that we have internally in the course, right? And so we write the paper in steps.
Mhm.
Um, and then you sit with other people who review your paper. So we divide the class into teams, and there's 9 teams, and then we take 3 of those teams and put them together into what we call a super team or a pod, and then you guys review each other's works. And so, can you comment then on the feedback and revision process and how that supported your development as scientific writers? And I will start with Sean.
Yeah, I really like the peer reviewing parts because it gave a time for other students to look at our paper, and then we could also look at their papers and share our perspectives. Um, and also we had uh, not only undergrads working with us, we also had graduate students, and they're more experienced as well. And some of them are doing research in structural biology, and so getting that type of expertise um, really helped in the making a more, I would say, a more professional research paper.
Okay, Gia?
Yeah, I definitely agree. I think as we're kind of it was nice having these like scheduled. I don't remember how often we had them, but it was definitely like often enough that every time we had some kind of progress that like we needed someone to kind of like bring us like back to the big picture. Because when we're just focused on like such a small portion or we're stuck reading the literature obviously all the time, or making even just something as simple as making a graphic, someone can give us a pointer to make our graphic way more clear to someone else who isn't doing what we're doing.
Okay, cool. Um, so how did working together as a team, how did that influence the quality of and depth of your work? Did you, how did you guys divide the responsibilities? Did you sort of go off, sort of play off one another? For example, Sean have a certain strength or Gia had a certain strength? And so how did you guys work as a team? And I'm going to start with Gia on that question and go to Sean.
Yeah, I think we generally work together. I think, Sean definitely has a lot more of the biochemistry knowledge and like the like as a strength, like, in comparison to me. And I think, that like that was like definitely like much more helpful for some of the like the structural portions and things like that. Whereas like, like maybe like I was making some more of the figures or something like that. But generally I think we just worked together throughout the paper.
Hmm.
Yeah, we worked together. We also did, yeah, we divided some sections up. So Gia did a lot of the introduction and the homologue section, and I did more of the interactions and the molecular mechanism
Hmm.
aspect of it. Um, but besides just splitting up, we also ended up overlapping a lot, and we just kept on making different drafts of each of our sections.
Hmm.
And so it was it was a very collaborative experience, I would say.
Hmm. Yeah, so if remind me, correct me if I'm wrong. So Sean, you're sort of have the biochemistry. You were getting your degree and specializing in biochemistry. And I think you're in the
Mhm.
chemistry department, specializing in biochemistry, or was it
It was in biology department. I did a chemistry minor though, and then
chemistry minor? Okay, but gee, I think you're a computational biology. Is that correct?
I'm actually just a plain biology
Oh, just plain
major.
biology
Yeah.
major. Okay, okay. All right, but then that sort of complemented. You're more of a generalist, where
Yes.
Sean was more a specific mechanism guy, right?
yeah.
That seemed like they were
Yeah.
complimented you guys complimented well, right? And so yeah, so certainly the paper was really high quality and really was very impressive with your work. Um, so um, so what was sort of most important skills or perspectives you gained for working on this project?
Mhm.
Um, Anyone can start, but I'm looking at G. I'll start with G.
Yeah.
Jump to Sean.
Yeah, I think you had a good point with the me being more of a generalist thing, but I think like, just the level of detail that is required for a project like this, like, applying everything, going back and, like we think we have it and then we read something in another paper that kind of like makes everything different. Practicing just that level of detail and attention to something, important, so important like this research paper was, I think, really good for me as someone who's like planning to pursue science, obviously. Um, and definitely, I think using all these different tools, it was very, very cool for me to like learn how to do basic coding and use all these novel tools to try to study this structure when I had never done something in the past, and this is something I hope, I'll be using in like research in the future.
Hmm. Sean?
Yeah, I would say the most important perspective I took from the class was how important just studying structural biology is for developing therapeutics in the future. And so, um, for drug discovery, you need to have the structure in order to uh, design rationally design a drug. And so, just getting that uh, getting that spark from the class really um, sort of inspired me to do this type of research.
Okay, cool. So you guys also, both of you guys have a really interesting perspective, right? So not only were you students, you were both TAs, right? And so I would be interested to hear your comments on being on the other side of this. Now you written your paper, you've done your thing, and then you paid it forward by coming back to my class and acted as TAs reviewing other students' work. So how was that experience? We'll just start with Sean and then go to Gia.
Oh, I enjoyed it. Yeah, I really like teaching, and so being able to have that opportunity, just being able to show other students uh, what steps need to be taken in order to write a decent paper, it was rewarding. I would say that so there are students that were also as passionate as me, and I really like that. And then also there are students that just wanted to do it for the requirement, and also that was also okay. But um, I would say that just being able to engage with those students from the other end, um, whether it's like grading papers or doing the nitty gritty of writing a paper, all of that was amazing.
Mm-hmm. Gia?
Yeah, I think so too, because obviously it was so rewarding to kind of see, you know, our, our research be put into, like, be written into this paper and everything, but like helping these students go from being super overwhelmed, kind of like how we were when we first received our structures, to kind of guiding them through just, step by step because the class is structured so well, and to where they feel like they could even explain it to someone else. I think it was just such a rewarding experience coming back and doing that.
Cool. So how has this course or this experience influenced your future career goals, Gia?
Yeah, I think this project has definitely really helped shape the way that I think as a scientist. So I'm going to medical school this fall. And obviously, there isn't any medicine without research, and research is something I, plan to continue pursuing. I actually, after doing this project, I did a different research project. It was just a summer project, but um it was more computer based and like more coding based, and because of I think the skills that I took from the analysis I did from here, I was able to apply just these ways of thinking to that project. And I think using these skills that I developed and kind of my mindset that I got from this project, this is something I'll carry with me. And also just such an appreciation for the most basic scientific research, especially in medicine.
Mm-hmm. Sean?
Yeah, I would say that it already has impacted my career. I just after graduating from WashU, I went to the National Cancer Institute and a Center for Structural Biology, and I did research there for a year. And so, yeah, I really uh, went deep into the deep end of the pool of structural biology with that experience. And I'm going to, like Gia, I'm going to a medical school, doing a MD PhD, and where I plan to do uh, more research in structural biology. So.
Mm-hmm. Well, okay. How about this? So, since I have you here, Sean, can you tell me a little bit about your research, as much as you can, about your research at the National Cancer Institute? Because you work in a structural biology lab, correct?
Yes, I worked in the Montero lab. It's in the National Cancer Institute, and yeah, I was responsible for studying this protein um, called DAP synthase or ARO3 from Canada auris, and Canada auris is another pathogen. It's a fungal pathogen, and so I was responsible for characterizing this uh, relatively understudied protein, studying both the structure. So it was a crystallography lab. Um, it was a high throughput crystallography lab. And so we used a national laboratory in Brookhaven, the synchrotron there, and we got a lot of structures for my protein. I also characterized it biochemically using isothermal titration, calorimetry, and differential scanning fluorimetry. And so, yeah, a lot of wet lab and dry lab in there.
Yeah, so you look at it from the other end, the structural biology, right? So it's certainly in my course, it was the end product was a structure, and then you did the analysis, and now you get to generate your own structure
Right.
from purification. So you've seen the whole gambit now. So
Yep.
how is that now? So now you're a structural biologist now, huh? From beginning to end.
Yeah.
So yeah, that's really cool. And so it looks like you guys are, so you guys are now, bachelor's degrees carrying scientists, and you've moved on. And so you had mentioned it before, but can you give me a little bit more details, Gia, on where you're going and what you're doing right now?
Yeah, I'm going to SLU here in St. Louis for med school. I obviously haven't looked at what research opportunities there are
Hmm.
yet, but um, I do plan to pursue research while I'm in medical school, and maybe after we'll have to see.
Hmm.
But of course, like I'm not super sure like where like what kind of doctor I want to be or anything like that. But I definitely have a very high appreciation for the basic science research.
Hmm. And how about you, Sean?
Yeah, I'll be going to University of Toledo. And yeah, like Jay, I'm not like committed to some specific subspecialty, but yeah, perhaps something related to hematology, oncology, or some internal medicine would be great.
Right. But you're going to pursue your MD PhD,
Yes.
Sean, and then
MD,
Gia.
PhD, yeah, yeah.
Right. And then Gia, her MD. Okay.
Mhm.
Great. So that's very impressive. You guys, and I will just add that you guys were, I was happy to have you in my class and was very impressed with of the work that you did. And so happy that you guys went on to be TAs for my course, and you've taken, it sounds like you've taken the experiences from the course to do other things. Some people stayed in it, stayed in the weeds, and some people just took the experience and applied it to the next thing, and both are great things. But yeah, I'm thrilled for both of you. You guys are going to be doctors someday. And then hopefully you guys will develop courses and have students and train the next generation of scientists, and so yeah, so I hope you guys enjoyed your experience at WashU. And so just one sort of last question. So what would advice you should give to a student that would take like my course or another type of course at Washington University? These, we call them, course-based undergraduate research experiences or cures, but these sort of higher level cure-like courses or these capstone-like courses, do you think they're worth it? And I'll start with Gia and go to Sean.
Oh yeah, I think they're definitely worth it. I mean, like where else are you gonna be able to apply like what you're learning like that? And I mean, the mentorship that you receive in a course like that is so valuable. We couldn't have done any of this without you, Dr. Smith, obviously. And I think like the advice that I would give is that whatever you want to get out of this class, obviously, is what you want to put in. Like what you put in is what you will get out of it. So the more that you put in, like the more that you will take from this class, and not just to do it for the grade, and really try to um, see what the bigger picture is.
Hmm. How about you, Sean?
Yeah, I would say that these types of classes are very good. To quote some physicist in MIT, he said, "It's not about what you cover in this class; it's about what you discover in this class." And so I think that applies a lot to structural bioinformatics. Um, and going off of what Gia said is, yeah, the more you put into it, the more rewarding it will be. That's especially true with this class as well. So what I would advise students who are coming into this type of class is to try to go all out in this class. Try to be as creative as possible, especially like sections like the what we call the wild card section, which is where you try to think of something unique about your protein. Try to try to uh, go into that a lot and emphasize that, and this will make your paper stand out and make the whole process very rewarding.
Great. So, yeah, I just want to say it was great talking to both you guys. It was again wonderful having you in my class. I'm so thrilled for you. You guys are, you guys are so young in your careers, and the sky's the limit. And I'm just excited for both of you. And good luck at medical school, and good luck at medical school and graduate school for you, Sean. And just keep in touch. I'd love to hear from from you guys.